Next, we move on to specification 2, which asks us to understand that the gene that controls the production of human insulin has been inserted into bacterial DNA. To understand this, we first look at the role of restriction enzymes. These enzymes act like molecular scissors, cutting DNA at specific recognition sites. When they cut, they often produce sticky ends, which are short single-stranded sections. These sticky ends are important because they can easily pair with matching sequences, allowing different pieces of DNA to join together during genetic engineering. As you can see in the diagram, restriction enzymes cut both strands of the DNA at precise points, creating these sticky ends. This makes it possible for a gene, such as the human insulin gene, to be inserted into another piece of DNA, like a bacterial plasmid. Now, in the production of human insulin, genetic modification is used in medicine to solve an important problem. The gene responsible for producing human insulin is taken from a human cell and inserted into bacterial DNA. Once this gene is inside the bacterium, it provides instructions for making insulin, which is the hormone that controls blood glucose levels. After the gene is successfully inserted, the bacteria begin to use it and start producing human insulin as they grow. These genetically modified bacteria are then placed in fermenters, where conditions such as temperature, pH, oxygen supply, and nutrients are carefully controlled to ensure maximum growth and productivity. Because bacteria reproduce very quickly, they are able to produce large amounts of insulin in a short period of time. Once enough insulin has been produced, it is extracted either from the bacterial cells or from the surrounding liquid. It is then purified to remove any unwanted substances or bacterial components, making it safe for medical use. The purified insulin is then used to treat people with diabetes, helping them control their blood glucose levels effectively. As you can see in the diagram, the process begins with the insertion of the human insulin gene into a plasmid using restriction enzymes and ligase, forming a recombinant plasmid. This plasmid is then transferred into a bacterial cell, creating a genetically modified bacterium. The bacteria are grown in controlled conditions, where they multiply and produce insulin. Finally, the insulin is extracted, purified through several steps, and prepared for medical use. This method provides a reliable, large-scale, and safe way of producing insulin that is identical to natural human insulin.